everyone. My name is Darla Saunders, and on behalf of the Atlantic Salmon Conservation Foundation and the Canadian Rivers Institute, I would like to welcome you to today's webinar. This series is made possible in part thanks to the contributions of the Government of Canada. For today's webinar, we're very fortunate to be hosting Mark Heaton. Mark is a biologist with the Ontario Ministry of Natural Resources and Forestry and is based out of the Aurora District Office just north of Toronto. He started with OMNRF in 1985. Mark is involved with species recovery, including peregrine falcon, Atlantic salmon, red-side dace, and brook trout. He has worked with many angling and conservation organizations with fisheries enhancement projects over the years, including fish culture, habitat rehabilitation, and fishway construction. After the presentation, we'll be opening the floor for a question and answer session. You'll have the option of asking questions directly using your microphone, or you can type them in and we'll read them aloud. I'm now going to turn the webinar over to Mark.
Thanks, Darla. Today's webinar is on Scotty boxes, experiences using in-stream incubation units for Atlantic salmon eggs and in-lake incubation units for trout eggs. Presentation outline is as follows. We'll cover off the Jordan Scotty box as well as other in-stream incubators, the Lake Ontario Atlantic Salmon Recovery Program and the use of in-stream incubation units, the Reserve Beauchene and the use of in-lake incubation of trout eggs, and cover off some of the lessons learned over the years that we've been doing this work. For those of you that are not aware of the Scotty boxes, they're referred to as the Jordan Scotty Salmonid Incubator, and they're manufactured by Scott Plastics out of Sydney, British Columbia. They were originally developed back in 1980 by Fred Jordan. Since that time, Scotty Plastics has taken on production of these plastic units. They're developed to enhance in-stream incubation of trout and salmon eggs, and they're affectionately known as the salmon condominium when it comes to the classroom hatchery program with the Atlantic salmon recovery work that we do in Lake Ontario. Two plates make up an incubator. These plates come in a variety of different colors that are associated with the hole diameter in the incubation plate itself. So red plates are a little bit smaller hole. They're used for coho salmon, pink and sockeye salmon, as well as trout species. The green plates have a larger hole and they're traditionally used for Chinook salmon. The yellow plates include a screened hole to contain fry and can be used for walleye. We've also had clear units produced in this style, and the local kids in the community classroom hatchery program refer to them as the fish jails. Fertilized green eggs or eyed eggs can be used in these incubation units. The eggs are placed on into individual cells with the assistance of the loading tray. The image on the right-hand side of the screen here has a fisheries technician placing the eggs into that loading tray with the assistance of a feather. 200 cells make up the unit, and typically they're deployed in blocks of 5 units or 1,000 eggs buried into the gravel on the upstream side of a riffle in a stream. The Vibert box was originally developed by fisheries researcher Richard Vibert of France and used in 1950. It was later modified by David Whitlock in the 1970s to include an egg incubation compartment. So when we're looking at this plastic unit here, this is the egg incubation compartment at the top, and it's got a plastic folding door, so you fill the eggs, put the eggs inside of that compartment. The intent here is for the eggs to hatch in that upper egg incubation chamber and drop down as sac fry into the protected nursery area, and then escape into the surrounding gravel as fry, and eventually emerge as swim-up fry. The design is now patented by the International Federation of Fly Fishers as the renamed Whitlock Vibert box. It's used for a variety of research projects, fisheries enhancement, and educational purposes. It's only available through the International Federation of Fly Fishers. Another one of the in-stream incubators that's been used over the years is the gravel-filled tubes, and it's been used in a variety of different applications. The tubes are made with a plastic or PVC pipe or rigid plastic mesh. Here we have a 4-inch ABS pipe with a cap on the bottom, and that plastic pipe is surrounded in the window screening, the fiberglass window screening. The bottom image is the screened mesh tube with the sock on the upper part of it for capturing the swim-up fry as they emerge from the gravel that's inside this plastic-filled mesh tube. Typically you're putting in between 100 to 150 eggs in these tubes. Moving on now from the incubation units and their different types, we'll move into the Lake Ontario Atlantic Salmon Recovery Program. The history of Lake Ontario is very interesting. Salmon, the Atlantic salmon, were one of the most abundant species in Lake Ontario prior to European settlement. The species was considered landlocked into Lake Ontario and used the North Shore as well as South Shore tributaries for reproduction. The first account of this species was recorded in 1656 by Jesuit missionaries. Later, as the country was settled, salmon were a very important source for food for early settlers in the 1700s and early 1800s. Dams were considered the first cause for decline of the species in Lake Ontario as a result of fragmenting the large lake from all the spawning habitat and nursery habitat in the watersheds that fed into it. The recovery efforts for Atlantic salmon actually began back in the 1860s with the construction of the first government hatchery built by Samuel Wilmot on Wilmot Creek, very close to Newcastle, Ontario. The species, unfortunately, was declared extirpated from Lake Ontario in 1896, with the last specimen just being caught off the Scarborough Bluffs outside of the Toronto area. Our recovery program for the Lake Ontario Atlantic salmon began in 2006. It's a cooperative recovery program led by the Ontario Ministry of Natural Resources and Forestry and the Ontario Federation of Anglers and Hunters. The goal of the program is to restore a self-sustaining population of Atlantic salmon to Lake Ontario and its tributaries, leading to a sustainable recreational fishery both in Lake Ontario as well as the tributaries that feed into it. We have a specific number of target streams that we focused in on for this recovery program. Of course, when you look at species recovery at the provincial level, when we do native species recovery, it's a fundamental action of Ontario's biodiversity strategy. The Lake Ontario Atlantic Salmon Recovery Program is built on 4 pillars: fish production and stocking, habitat and water quality enhancement, research and assessment, education and outreach. These actions go through and are assisted by a number of program partners, and we have over 40 different program partners involved with the program now. We work with 3 different strains of Atlantic salmon for recovery. We have the Sebago Lake strain from Maine, the La Have strain from Nova Scotia, and the Lac Saint-Jean strain from Quebec. Our adult returns were first detected in local fishways beginning in 2009. This map depicts the target streams that are associated with the Atlantic Salmon Recovery Program. The dark blue streams represent those that are being stocked with fry, fingerlings, and yearlings. The light blue streams are those that are focused on for habitat rehabilitation. So currently we have a number of different life stages of Atlantic salmon and 3 different strains being stocked into Cobourg Brook, Duffins Creek near Pickering, and the Credit River as it flows through Brampton and Mississauga. Our adult brood stock are held at the Ontario Ministry of Natural Resources Harwood Fish Culture Station located on Rice Lake. There they produce the eyed eggs that are shipped to other facilities, including the Normandale Fish Culture Station, which produces a large number of advanced fry for stocking in May, fall fingerlings that are stocked in October, and yearlings that are stocked in April. Community hatcheries run by volunteers are provided by eggs which raise the fish to fry stage and release into local target streams. And we have a number of clubs associated with the Atlantic Salmon Program, including the Credit River Anglers Association, the Islington Sportsman's Club, Metro East Anglers, and the Bell Fountain Community Hatchery. Over 100 classrooms across the Greater Toronto area raise fry from eyed eggs, and they release those fry into local target streams. The surplus eyed eggs that we get from the provincial fish culture system are also used for research and stocking. We'll now move on to how in-stream incubators are used with the Atlantic Salmon Recovery Program. Since 2008, Ontario Streams, one of our key partners in the program, has been involved with in-stream incubation with eyed Atlantic salmon eggs provided by the Ontario Ministry of Natural Resources and Forestry. Whitlock vibrant boxes, Scotty incubators, and gravel-filled tubes have been used in comparison with direct deposit and injection methods for eyed eggs. The investigations thus far have focused primarily on the upper Humber River, just north of Toronto. The Humber River is known historically as one of the important Atlantic salmon streams feeding into Lake Ontario. Incubating Atlantic salmon eyed eggs directly in headwater streams is seen as a potential tool for restoration when it comes to this program. So they've been looking at the different techniques to see which ones work best. What they use as an indicator is the production of fry and fingerlings, and the survival at this stage helps them evaluate which ones perform better in terms of methods. Early in the investigation, there were 6 incubation methods that were tested. Scotty boxes with screens that trapped the fry in position, and those Scotty boxes were placed in the gravel. Scotty boxes without screens, and again, they were placed in the gravel. The Whitlock vibrant boxes placed in the stream directly in the gravel, direct deposit of eggs into gravel, and then broadcasting the eggs into the current of the stream. The work with the eyed eggs was done in the January to early February period, with fry retrieval in May and fingerling surveys in late summer. This is the Upper Humber River. The watershed is about 900 square kilometers in size. The area is basically 30% forest cover, with the dominant land uses associated with agriculture being hay, as well as livestock production and some intensive row crops. The western part of the watershed is having the Niagara Escarpment passing through it, which is a significant source of groundwater to the system. And then further to the east part of the Upper Humber watershed is the Oak Ridges Moraine, which is a large sand and gravel deposit that also provides cold water to the tributaries. The work that we're going to talk about now with Ontario Streams is focused on some private properties in this area, being the Coffee Creek Farm, the Stewart property, and the Finest property. If you're interested in looking on a map, the area is just located west of Highway 50 near the village of Palgrave, Ontario. This is the 2008 results of the incubation study that was performed, and they looked at a number of different methods, including the direct deposit into gravel, Scotty boxes with and without screens, Scotty boxes placed in the gravel as well as above the gravel, tied into the bed, the bank of the creek, the Whitlock vibrant boxes in gravel. They even tested out some of the Whitlock vibrant boxes in a controlled environment in the community hatchery. Unfortunately, the percent survival of stocked eyed eggs to fry stage was relatively poor. The best return was on the Rogers Creek Scotty boxes, which is a different watershed with low sediment. And basically they got 49% survival for the 600 eyed eggs that were placed into those incubation units. Again, looking at the late summer fingerling production, the numbers here are very low in terms of number of individuals caught per square meter of stream. Now, this was replicated in 2009, and similar results were found. So based on the poor outcomes of 2008 and 2009, the methods were refined in 2010 based on the following things they learned. Scotty boxes and Whitlock vibrant boxes without screens could not be monitored for effectiveness in producing fry. There's no way to count what comes out of these structures. The direct deposit method of putting the eyed eggs into the current over cobble produced no fry. The method of using screened Scotty boxes was the focus of 2010, with also adding on the additional method of using screened gravel-filled tubes. Just go through a series of photographs showing the deployment of the Scotty boxes. So here we've got that technician that's providing some assistance in getting those eggs into the 200 cells of the loading tray. Once the loading tray has been finished off and checked for quality, the next step is to put the screened Scotty box panel on top. It is then inverted, and then the second unit is placed on top. Once the two panels are placed together, they're secured using a nylon nut and a stainless steel bolt. Or sorry, a nylon bolt and a stainless steel nut. The Scotty box is then placed in the stream, and the image on the left here shows that Scotty box. This one's actually placed above the gravel and anchored to the bed of the stream, and the position of this one is referred to as being parallel with the current. The image on the right is showing the sediment accumulation in that Scotty box following retrieval of that same unit in May. These are the plastic gravel-filled tubes. So here we've got the 4-inch ABS pipe with a cap that's solvent welded on the bottom. The mosquito mesh or window screening is affixed to the side using glue. The tube is filled with gravel. It's typically 1 1¼ inch diameter gravel. You can see a few eggs in this unit on the right-hand side. The eggs are counted into the tube. Once the tube is backfilled with gravel and capped, it's placed into the bed of the stream on its side. And here, the image on the right, you can see that tube on the bottom placed in the cobble of the stream, and there's a piece of green flagging tape attached to it, and there's a good purpose for that. When you look at the next image on the left, the gravel tube has been covered in cobble, and now all you have left is the flagging tape sticking out, flowing in the current that tells you where your units are, especially when you come back to retrieve them 3 months later. So the tubes are retrieved in May, and it's usually the first or second week of May, and we do keep track of the water temperatures to help us predict the best time to pull these things out of the creek. The tube is opened and the fry are counted, and it's both live and dead fry that are counted. So as a result of the 2010 work, with 16 Scotty boxes, they found that there was a varying survival rate of between 0.5 to 10.8%. The sediment accumulation in the Scotty boxes was the primary reason for reduced survival. There was little difference in survival when you compared the Scotty boxes in relation to whether the box was parallel or perpendicular to the current. Now, when we look at the 18 gravel-filled tubes that were used, it resulted in a 24.5% survival to swim-up fry stage. Moving into phase 4 of the investigation in 2011, Ontario Streams focused on the method of using screened gravel-filled tubes based on the performance from 2010. This phase included using different densities of eggs per tube, and they upped the numbers of tubes from 18 to 250. They also solicited the help from PhD students from McGill University to analyze the survival data based on egg density. This is the results coming from the PhD students. So we've got 6 different properties here. Chico's, which is located at Highway 9 on the main Humber River, had very poor survival based on the 3 different scenarios of 50, 75, and 100 eggs per tube. Coffee Creek Farm had moderate survival of about 40% for the 50, 75, and 100 egg densities. The Galloway site and the St. Francis site had much higher survival for those eggs at those different densities, and they also looked at density of 150 eggs per tube, and then they had one for 300 over here. And what they found was that this situation, these locations were the ones that were directly downstream of online ponds. The 2011 survival to swim-up fry stage ranged from 2.5 to 80.3% using these gravel-filled tubes. The survival did vary between sites, and again, the highest survival occurred where the incubation tubes were located downstream of online ponds. In 2012, they continued with the use of the incubation tubes and then also adopted the in-stream incubation system method developed by the Department of Marine Resources in Maine. This method uses a gas-powered water pump and plastic funnels to inject the eyed eggs into the gravel substrate. Survival to fingerling stage was very poor using this technique. the highest fry survivals observed in 2012 were based on the plastic tubes that had an egg density of 150 eyed eggs per tube. We'll just show a few photographs now of the incubation tubes being placed in the stream. So the image on the left-hand side of the screen is the channel that's been modified. The volunteers will rake out a trench and place the gravel tubes in the trench on their side, and then backfill with the cobbles over top. And then following in May, those gravel tubes are extracted and the fry are counted. So we've got some of those Atlantic salmon fry in the image on the right-hand side of the screen. There's a lot to this image here that I need to explain. This is the in-stream injection system, and there is a video available on YouTube called Penham River Egg Injection. The gas power pump that is used is here on the bank of the stream. It's a 1-inch gas powered pump that is connected to a garden hose. The garden hose is connected to this steel pipe. This steel pipe is placed inside a funnel, so we've got a 4-inch segment of PVC pipe. Underwater, we've got a 4-inch to 1-inch reducer going to a 14-inch segment of PVC pipe. It's cut on an angle. These funnels are driven by water injection down into the gravel of the stream. So here's a funnel that's in the correct position for depositing the eggs, and the technician is counting out the eggs and placing them into that funnel. Once the eggs settle into that 1-inch tube, it's very cautiously pulled out, and the eggs are left behind inside the gravel substrate of the riffle. When we look at the 2012 results, we have 3 different sites here. So we've got the Galloway property, which is just directly downstream of an online pond. The different scenarios that were run here were 150 eggs per tube up to 500 eggs per tube. The number of eggs here we're working with is approximately 18,000 eggs. The percent survival for Galloway using the 150 eggs per tube was 55.8%. They got 60% with the 300 eggs per tube scenario. St. Francis, again, directly downstream of the online pond, 71% with 150 eyed eggs. With the 300 eggs scenario, 58.1%. The St. Francis site is, this third site is actually upstream of the online pond, and you can see here that percent survival to fry stage was much less. When we look at the August electrofishing results, where the technicians will go back into that same reach and electrofish for juvenile Atlantic salmon, they'll be counting all of the fish captured that are under 100 millimeters as summer fingerlings. So in this case with Galloway, they captured 31 juvenile Atlantic salmon and 1 older fish from probably the year before. Their Atlantic salmon individuals per square meter was 0.2, which is a good return rate, we believe. As compared to the St. Francis Center, which had poor returns, when you look at the Atlantic salmon produced per square meter, it's about 1/10 of that. So there's an important message here with the implications of online ponds. Typically we don't think of them as very good, but in this case they are helping out with Atlantic salmon incubation in the tubes by holding up some of the fine sediment that's moving through the system. Within the 2012 work, they also looked at a 5-year synthesis of the results. So here we have the legend at the bottom, just describing the different scenarios, different methods. So we've got Scotty boxes. The best fry ever seen for that 5-year period was the Scotty boxes in the Upper Humber was 10.8%. The Scotty boxes in the Credit River, Rogers Creek, best seen for fry production was 49%. And then the best fry production overall was with the in-stream plastic tubes, with 71% fry production being the best performance. When we look at the maximum average survivorship of the eyed egg incubation methods for the month of August, using Atlantic salmon per meter squared as the indicator here, our best performing situation was the plastic tubes, gravel plastic tubes, with 0.2 Atlantic salmon fingerlings per square meter. And just as a comparative, we've got Islington Sportsman's Club fry stocking, where they'll produce 20 to 25,000 fry. And in those areas where we go back and monitor the site for fingerling production some 4 months later, in that area they were producing about 0.1 fingerling per square meter. So now move to the 2013 locations. So in 2013 they started focusing in on these online ponds. So again, we're in the Upper Humber. We're just north of Highway 9 with the St. Francis Center. They've got an online pond here. They looked at 2 locations, one directly downstream of the online pond, one located about 200 meters downstream. Same with the Galloway property here, directly downstream of the structure, the online pond, and then one located a bit further downstream. And then we've got another private online pond here. It's actually a micro-hydro facility in the Upper Humber, and they've got some of the incubators located directly downstream of the structure, and then a few hundred meters further downstream. So in terms of the spring 2013 fry survivorship, using that incubation tube stocking sites with these different scenarios involving online ponds, what they found was that as you go further downstream for the dam, the survivorship was actually less. So here we've got the results of the online pond of the main Humber, close to the dam, roughly 25% survival, but as you move downstream, the survival dropped off. Here, the St. Francis Center again, 43% survival directly downstream of the structure. Survival's diminished as you move downstream. And with the Galloway property, similarly, but still good survival on both scenarios with that online pond. So between 2008 and 2013, 7 incubation methods were tested. They tested Scotty boxes with screens, with them placed directly in the gravel. They used the Scotty boxes without screens directly in the gravel. They also used Scotty boxes placed above the gravel and anchored to the bank of the stream or into the woody debris of a pool. The Whitlock vibrant boxes were used in gravel incubation. They also looked at direct deposit of eggs into the gravel, broadcasting the eggs into the current of the stream with the intention of having those eggs settle into the interstitial spaces of the cobble. And they also tried out the in-stream incubation system. The key findings here is that the screened gravel-filled incubation tubes were the most effective in producing spring fry. The screened Scotty boxes were moderately effective where in-stream sediment loading was low, being the case with Rogers Creek and the Credit River. Deploying in-stream incubation units below online ponds improves the survival of eyed eggs to the swim-up stage. If you're interested in learning more about this, you can go directly to the Ontario Streams website, and they've got a page there entitled Studies, and you can download all of the technical reports of the past 5 years. Those reports go into a lot more detail than what I've just covered off here. Now moving into the component of the Atlantic Salmon program for Lake Ontario, we've got a lot of interest in getting people involved. So we have a good outreach and education program, and one of the big pieces of that is the classroom hatchery program that we do with the Ontario Federation of Anglers and Hunters and local schools within the Greater Toronto area and beyond. So this is a very important part of our recovery program. And in it, we use the Scotty boxes in the classroom hatcheries for students to experience the development of the eyed eggs into free-swimming fry. There's no better structure to cover off having a kid watch a fish hatch from an egg right in front of them using the Scotty box. This acts as a learning conduit for teaching biodiversity, water quality, math, and natural history. So in 2007, we began the program with 21 classrooms across the Greater Toronto area. And as of 2013, there were over 100 schools and outdoor education centers involved, including the Toronto Zoo. We had 1,325 students engaged in 2013, and we're expecting more in 2015 with over 110 schools ready to receive eggs over the coming weeks. Just a few photographs showing what this is all about. So we operate with 100 eyed Atlantic salmon eggs, and here we have Chris Robinson from the Ontario Federation of Anglers and Hunters. He's our coordinator for the Atlantic Salmon Program, and he's loading up those eggs into the loading tray, which will be moved into the clear plastic screened Scotty box. The eggs are transferred into that Scotty box and then secured, and then they're placed inside the classroom hatchery. And the classroom hatchery consists of about a 25-gallon aquarium, and the aquarium has a small pump in it that pumps the water out to a chiller unit, and it's usually a 1/10 horsepower chiller unit that we get from Big L's Aquarium Services in Toronto. There's a biological filter that's also tied in with the classroom hatchery. Here's a complete system here. So we've got the chiller unit at the back, the biological filter here. The aquarium is wrapped with insulation to help conserve energy, and we've also got the program materials here for the Atlantic Salmon Recovery Program, including life cycle information and more information about the history of the species, as well as all the project partners that are helping out with recovery efforts. Once the eggs have developed into free-swimming fry, they're released from the Scotty box, and then they're transported by the kids to the local target stream and released. Typically this is happening in the last few weeks of May and early June, and we'll get the entire classroom out with the teacher, learning about the stream, learning about benthic invertebrates, and helping with the cleanup of the stream and releasing their fry. And in terms of survival, we've had a variety of different scenarios with the program. We've had zero survival when it comes to these units as a result of technical errors. Somebody pulled a plug and the aquarium got too warm. And then we've had situations where they've had 90% survival to swim up stage. So it's really been helpful having the Scotty boxes as part of the classroom hatchery program because it enhances the survival to the swim up fry stage as compared to traditionally just putting the eggs into the gravel of the aquarium. And when you position that clear plastic Scotty incubator up against the glass of the aquarium, it allows the kids to see everything. They can see the eyes, they can see the eggs hatch right in front of them, they can see the little alevins wiggling away in the compartment, and gradually as that yolk sac absorbs, they can see the coloration changes happening with the fry. So now we're going to move over to the work that's been done at La Reserve Beauchesne in Quebec. For those of you that are not familiar with La Reserve Beauchesne, it's about a 50,000 acre privately operated fishing and hunting destination in Temiskaming, Quebec. It supports 40 lakes, with the largest lake being Lac Beauchesne itself, which is about 15 miles long. It supports a variety of species, including walleye, smallmouth bass, northern pike, whitefish, lake trout, and brook trout. Many of the brook trout lakes that they have are managed for put-and-take fisheries and are stocked annually under license issued by the Quebec government. 4 of the lakes that they have are managed as wild no-kill fisheries, and these are self-reproducing brook trout that rely on one spawning shoal for that entire chain of 4 lakes. The past history of gravel extraction upstream of this gravel shoal has resulted in diminished reproductive capacity for the chain of lakes. This is the flashing yellow here is the Taggart Lake, now known as Watrous Lake in memory of Richard Watrous, one of the key people that started up La Reserve Beauchesne. He passed away a couple of years ago. This is the main Lake Beauchesne here, which is about 15 miles long. I'll tell you a little bit more about this work. Basically, we do the egg collection here for the adult brook trout, and then we incubate the eggs in the main part of the lake close to the outlet. So this outlet drains into the Beauchesne River, which eventually goes into the Ottawa River, and it's a good-sized river with a high gradient. The use of the Scotty boxes began in the fall of 1997 when Reserve Beauchesne was looking into ways to improve the Taggart or Watrous Lake brook trout population. The fertilized green eggs were collected from that Watrous Lake brook trout and incubated using the boxes, and the boxes were buried into the spawning shoal in gravel adjacent to the redds that had been dug by the adult fish. The boxes were retrieved the following May and found to have limited success. And there was problems with freezing. If you're not directly over top of that trickle of groundwater coming into the shoal, your eggs will freeze. And there was no way of really detecting what your fry production was, because if they did manage to survive, they would escape into the surrounding gravel, and there's no way to count them. So over the years, the methods were changed to improve survival and maximize production. We began with collecting more eggs. So traditionally, we get a permit from the Quebec government for collection of 12,000 brook trout eggs, and we use 30 screened boxes, the screened Scotty boxes. They're set up as 2 eggs per cell with 400 eggs per box. The incubation occurs in Lac Beauchesne itself in 7 meters of water at the outflow of the lake where it transitions to the Beauchesne River. 10 incubators are secured to a 4-meter-long vertical PVC pipe held in place with a rope, an anchor, and a float. And there's a lot of time spent trying to figure out the best way to incubate these in a lake environment. We did try incubating at the inlets to the lake coming from the surrounding creeks, and we found that there was a lot of organic sediment moving into the incubators over the course of the 6-month incubation period. And we found so far that the incubation at the outlet of the lake is one of the best performing situations. One of the considerations that you need to keep in mind is that wave action, you want to keep that to a minimum. So using a small bay that's sheltered from the predominant westerly winds is important. We've also gone into looking at lake trout eggs. So we've been fertilizing some lake trout eggs based on collections out of the main Beauchesne Lake and incubated with the same Scotty boxes. So just go through a few photographs here, and a lot of this work is really based on volunteer assistance, and our primary volunteer that's been helping out, volunteer organization helping out, has been the Metro East Anglers. So they come and help with the collection of the adult fish and the collection of the eggs, setting up the eggs into the incubation units, and the fry release in the spring. So here we have one of the volunteers loading the Scotty loading tray, 2 eggs per cell. In this case, it's the lake trout eggs that are being loaded, and he's using a turkey baster, which is really helpful for controlling the eggs as they go in 2 by 2 into each of these little cells along this line here. And then if sometimes you have 3 pop out, 4 pop out, use the feather here to help move those eggs around. Here's a completed loading tray, and this is the older loading tray that was available back in the mid-2000 period. And again, it's lake trout eggs, and you can just see the feather being used to manipulate a single egg. So this incubator will be set up with 400 green eggs. It takes quite a few volunteers and quite a few nights of hard work to put 12,000 eggs into 30 incubation units. So we typically have a team of volunteers of 6 to 8 individuals. So here you can see that they're placing the eggs into the loading trays using the turkey baster here. We used ketchup bottles in the past, and we've got the feathers that are used for moving the eggs around. And then we've got the screened green Scotty box here, which will be used for transferring the eggs into once the loading tray is complete. The next morning, the Scotty boxes, well, basically they're held overnight in a cooler full of water. They're never left out of the water for very long. The next morning, we take them out to that bay that feeds into the Beauchesne River, and we position ourselves in that 7 meters of water and attach the Scotty boxes to the PVC pipe at about 30 to 40 centimeter increments. And we use the nylon zip ties for that. We found that they're very effective over a 6-month period in terms of staying in place and not breaking. We haven't lost any incubation units as a result of using the PVC pipe and the zip ties. We have lost units in the past as a result of the flotation. So it's an important consideration when you're using a flotation in this scenario, such as a Javex jug or an antifreeze bottle, is to rinse it out, clean it, and then fill it with the expansion foam and then cap it. Because 6 months underwater with these flotation incubation devices, you'll find that they do fill up with water and they can sink. That's not very good retrieving a set of incubators from 7 meters of water. So here we have the incubation units set up. We've got 2 strings here set up. So we've got some of those yellow units that are in place, and we've got some of the green units with the screen on. And here we're using the braided poly rope. It's really good knot strength. We've never had any knot failures. The first year we did it, we used some of that yellow polypropylene rope, and we had some issues with that. So one of the recommendations is to go with that white braided poly. And interesting, keep in mind is that in the fall, October, you're placing these strings in position, and you're placing those flotations so they're just below the water level. And the reason for this is that the lake is lowest in the fall. And when you come back in May to retrieve your incubators, the lake level can be 2, 3, 4 feet higher, just as a result of spring snowmelt. So we have these floats that are always close to the surface, within 3 or 4 feet, and we managed to grab them with either a rake or some other type of extraction tool. And from there we take them back, disconnect the nuts and bolts, and then start counting the fry. So here's a Scotty box, and you can see it's got the mosquito window screening attached to it. And then we've got a couple of brook trout fry. So in a really good situation, you're producing quite a few fry. In this case, we're probably a couple hundred fry that are produced out of this one incubation unit. You can see that the dead eggs and dead fry that didn't develop are here. And you can also see a little bit of saprolegnia fungus around the eggs. And what we've been finding is that where you do get a fungus outbreak, it can overtake even live eggs during the development period, and you can lose an entire Scotty box as a result of the fungus. So one of the recommendations is to always disinfect the units before you use them the following year. Once the fry are released from the egg incubation and counted, we then take them back to the Watrous Lake and release the fry into the same habitat as where the native fry, naturally produced fry, are being produced. So this is near shore with lots of cover from the cedar trees that have fallen into the shoreline. And it's just a picture here showing the brook trout fry, and they'll be feeding in a matter of a couple days. So in terms of the results from the work at Reserve Beauchesne, we found that the average brook trout survival to fry stage for the past 5 years is about 12.1%. Keeping in mind we're using green eggs, and between the time of setup to time of retrieval is 6 months in the lake. The survival ranged from 0 to 71% within the individual boxes. We have had really good survival for lake trout in the 3 years that we've done it. So the average survival to fry stage for lake trout for that period of 2010 to 2012 is 26.7%. And one of the biggest issues we've encountered with this technique is the fungus development during the incubation period. Just going over some of the lessons learned. So screened gravel-filled plastic tubes perform very well for incubating Atlantic salmon eyed eggs in the stream. Performance is better when the tubes are located below the online ponds, and this is within 10 to 20 meters of that online pond. As you go further downstream, the performance diminishes. The Scotty boxes can perform moderately well in streams where fine sediment loading is low. The Scotty boxes can also incubate green, fertilized brook trout and lake trout eggs in a lake environment, provided there's sufficient current during the incubation period. That current is really important. Some of the practical tips that we'd like to convey to those people that might be interested in this technique in the future is to thoroughly clean and disinfect the incubation boxes before use. Fill the floats with expansion foam. Nylon bolts and stainless steel nuts are best for securing the Scotty plates together. We have used different nuts and bolts and always had problems with either rust or the bolts coming undone during the incubation period. The braided white poly rope and nylon zip ties are the best way to secure the incubation units to the vertical anchor line. And make sure you locate those incubation strings where there's no wave action or very little wave action. I'd like to thank the Ontario Ministry of Natural Resources and Forestry for their support over the years with working with the different types of incubation units and the Atlantic Salmon Recovery Program. Our partners with the Ontario Federation of Anglers and Hunters have been instrumental in getting the program moving along at a very good pace with lots of different partnerships. Ontario Streams is an awesome stewardship organization that we work with, and they're the ones that are out there on a minus 20-degree day putting the Atlantic salmon eggs into the stream. The Metro East anglers have been very helpful providing volunteers with the work at Reserve Beauchesne. We also like to thank the folks at La Reserve Beauchesne in Temiskaming, Quebec. That's it, and thank you very much. And if you have any questions, I can take those now.
Thank you so much, Mark. That was excellent. A really great overview of the various programs and the successes and challenges that you've had. It's quite impressive. Thank you.